Hi, I'm Sophia Papakostas,
And I'm Denise Arroyo.
and we're presenting metabolomics and mass spectrometry imaging reveal channel de novo purine synthesis in cells. A metabolin is a multi-enzyme complex that is formed between enzymes of a particular metabolic pathway, which is held together by non-covalent bonds. The formation of metabolins enables intermediate products and substrates from one enzyme to be passed directly to the next sequential enzyme in the pathway to increase efficiency. The Purinosome is a multi-enzyme complex indicated by the sun in the image above, composed of 9 enzymes that are responsible for the conversion of phosphoribosyl pyrophosphate, PRPP, to inosine monophosphate, IMP, and then converted to either AMP or GMP. Past studies have shown that purinosomes are located in the cytoplasm near mitochondria or residing on microtubules and show directed movement towards a mitochondria. The channel pathway is indicated by the close association of the purinosome with the mitochondria, as the input metabolites needed for purine synthesis, such as glycine, aspartate, and formate, are derived from the mitochondrial metabolism. The unchanneled pathway is a pathway in which each enzyme works individually to form its products and obtains its substrates from the cytoplasm. This paper supports the hypothesis that cellular metabolic pathways organize in space and time to confer different properties on the collective versus individual enzymes, meaning that the enzymes that catalyze the de novo purine synthesis, DNPB, may localize together for different advantages or properties that are unique to the complex than to the individual enzymes. The null hypothesis was stated to be that the 10 DNPB steps that form IMP from PRPP are completely independent of each other, equilibrated with their cytosolic pools, and have a homogeneous distribution throughout the cell. This would mean that each step of the pathway is not affected by the other steps, as they are performed separately. The substrates are obtained from each respective cytosolic pool. And lastly, that there is a homogeneous distribution of these catalytic enzymes throughout the cell, showing that they are not localized together in a purinosome or near a particular organelle. The image shown on the left shows the DNPB pathway and the salvage pathway. The image on the right shows that each step, as well as the metabolites that are derived from the mitochondria. In this study, the researchers used purine-depleted HeLa cells, which show high DNPB flux, as a model system to probe the metabolic consequence of purinosome formation. These cells were grown in cultures containing isotopically labeled carbon and nitrogen in serine. The cells uptake the labeled serine from the media, which is internalized by a mitochondrial serine transporter, which is then metabolized to form formate and glycine. These metabolites exit the mitochondria and are used in a DNPB pathway. The researchers developed a model to mathematically predict the distribution of isotopic labels that would be expected in the absence of the DNPB metabolon, and then tested the predictions by performing an in vivo isotope incorporation experiment. As shown in figure 1e above, the plus 1, 2, 3, 4, and 5 correspond to the estimated probability of those given numbers of isotopes presented in IMP. Two assumptions must hold true. IMP, the two-ring structure above, and all the downstream nucleotides must have the same isotopomer distribution of plus 3, 4, and 5 species, and that labeled glycine and formate enrichment in the DNPB intermediates and N nucleotides should be the same. They quantify the isotopic abundance using liquid chromatography and mass spectrometry. Gas cluster ion beams secondary ion mass spectrometry permits high mass ion detection with low chemical damage to study the cytoplasmic distribution of intact molecular ions of purine biosynthetic pathway intermediates and N nucleotides in a frozen hydrated monolayer of HeLa cells. The instrument generates an array of mass spectra for each ICAR voxel 3D pixel in order to study their spatial distribution reliably and verify their identity. In contrast to the assumptions made by the model, the fractional isotope abundance in plus 3 and plus 5 IMP significantly differed from those in GMP and AMP, as seen in A. This shows that IMP and AMP/GMP have distinct isotopomer distributions. To understand this difference, they used the observed distributions of the IMP precursors FCAR and SICAR to compute the predicted isotope distribution of IMP using their model and compared it to the nucleotides downstream of it, GMP and AMP. As can be seen by figure B, IMP matched the model predicted distribution of isotopes, whereas AMP and GMP showed significant differences in their isotope abundance relative to the model, as seen in figure C and F. This model was predicted under the assumption that the isotope incorporation in all of these species would be the same if the substrates came from the same cytosolic pool. From these results, IMP and its precursors must have been synthesized using the same cytosolic pool of substrates, while AMP and GMP use a different pool for their synthesis. Moreover, no other intermediates showed the same isotope enrichment as GMP and IMP.
In the next experiments, they used the observed isotopomer distribution in AMP and GMP to compute the percentage isotopic enrichment in the source substrates of the channeled pathway. Compared to IMP, the newly generated AMP and GMP had a 10% higher isotope enrichment, higher glycine isotope enrichment, and 15 to 20% higher formate isotope enrichment. They concluded that the synthesis of AMP and GMP is accomplished in a highly channeled manner, preventing pathway intermediates from equilibrating with their bulk cytosolic pools, whereas IMP is formed in an unchanneled manner. The higher isotope enrichment in AMP and GMP relative to IMP indicates the physical proximity of an active DMPB metabolon or association to the mitochondria. To test this explanation, they treated the cells with a guanine synthesis inhibitor, MPA, which inhibits IMP dehydrogenase. This resulted in accumulation of IMP by a factor of 12, as can be seen by figure F. This occurred as a result of forced release of IMP synthesized by the channeled purinosomes, leading to intermixing of IMP produced from the two independent pathways. Consistent with the hypothesis, upon MPA treatment, the observed labeled formate enrichment in AMP and IMP was similar. To see if the DMPB metabolon association to the mitochondria increased efficiency, they knocked down the synthesis of 10-formyl THF by targeting MTH FD2 using small interfering RNA. Upon treatment, there was a disruption in the pathway efficiency by a factor of 100, reflected in the accumulation of the intermediate ICAR and a decrease in the production of the end product, AMP, relative to the control. This change in efficiency can be seen in Figure K. Figure J shows the proposed pathway of DMPB in proximity to the mitochondria. These observations indicate that the association of the purinosome to the mitochondria represent an active metabolon. The association allows for the capture of serine and formate from the mitochondria for AMP and GMP synthesis. This represents the channeled pathway where there is a limited equilibration of the intermediates. On the other hand, IMP arises from a diffusive substrate pathway or an incomplete purinosome. GCIB SIMS was used to capture purinosome assembly. This ensures quality resolution given that GBIC scanning uses a 1 micrometer focal diameter and purinosomes range from 0.2 to 0.9 micrometers. The expected results for an active DMPB metabolon would show a higher concentration of isotopically labeled intermediates and end product nucleotides in confined areas. Pictured here is the spatial distribution of the isotopically labeled 15N ICAR in one layer of the studied HeLa cells. A total of 2 or 3 layers from all the cells were analyzed independently. Figure C shows a zoomed-in region of the cell where you can better see the white ICAR pixels. These images of ICAR show a non-homogeneous distribution with distinct isolated higher concentration voxels, as well as a higher abundance of ICAR molecules per voxel by a factor of 300 to 1,000. From those previously captured layers, the isotope enrichment levels were compared among the DMPB intermediate ICAR and the N nucleotides AMP, GTP, and ATP. The ratio of labeled 15N to unlabeled carbon molecule was determined. Looking at figure G in each layer, the 15 nitrogen to carbon ratio for ATP was consistently higher in the labeled ICAR pixels than in the random pixels. This phenomenon confirmed the expectations that the enrichment was specifically signaled by an active channel DMPB pathway. However, the same trend was not seen for AMP and GTP due to proposed technical limitations. Moving on to figure H, a similar procedure was followed, except one of the de novo pathway enzymes, ATEEC, was knocked out. 15N enrichment for ATP was not observed in the ICAR pixels. This supports the idea that enrichment only occurs by instruction of an active channeled metabolon. Lastly, figure I displays the negative control, which utilized high glycine concentration. This environment provided inefficiently channeled DMPP pathways and limited 10-formyl THF led to decreased purinosome-mediated synthesis, which ultimately led to low ATP enrichment in the ICAR cells. All of these results support the hypothesis that purine production occurs in concentrated hotspot areas near the mitochondria. It was found that purinosomes Where's the conclusion?
Oh,
Oh, there. I'll edit this. It's okay. It was found that purinosomes comprise 9 enzymes that act synergistically, channeling the pathway intermediates to synthesize purine nucleotides, increasing the pathway flux and influencing the AMP to GMP ratio. Another key takeaway is that unchanneled pathway synthesis was found to favor guanine production, whereas channeled pathway synthesis was found to favor adenine production through different enzyme orientation. This supports the initial hypothesis, which states that cellular metabolic pathways organize in space and time to confer different properties on the collective versus individual enzymes. Future implications of this study include better understanding of purinosome activity, which may be helpful in developing therapeutic treatments for cancers that have high purine demands, as understanding the structure and function will allow us to target the vulnerabilities of the complex.